if we look at the process of aging from the perspective of protein misfolding aggregation, so not from a generic perspective, but only from the perspective of protein misfolding and aggregation, we have a situation like this one. When we are young, we have formation of protein oligomers, aggregates, misfolding, and so on. But these are counteracted very effectively by our proteostasis network formed by the translation machinery, chaperones, autophagy, and the ubiquitin proteasome system. And this proteostasis network can counteract very effectively misfolding and aggregation, and apparently we don't have any problem. But when we are old, then the situation changes, because we have still formation of oligomers, aggregates, and these accumulate in the human body, but these are no longer counteracted very effectively by the proteostasis network, because as we age, these lose in efficiency. So our chaperones, our autophagy system, our ubiquitin proteasome system works less effectively. And so we have a problem, actually. This is also a consequence of mutations. And as a consequence of mutations, we can have early onset diseases because of the balance, because of an early change of this balance. And so from this perspective, let me just emphasize once more that this is only from the perspective of protein misfolding aggregation. Aging is a progressive increase of misfolded aggregated proteins and failure of proteostasis. And as we heard from David Smith, this also leads to neurodegenerative diseases which are associated with aging. But the picture is more complex, because we don't have only a loss of proteostasis, we don't have only formation of oligomers or aggregates. We have a number of factors which are responsible for aging, and these participate to this complex scenario. This is a picture that I'm sure you have seen in slightly different forms in many circumstances. This lists all the possible conformational states for a protein and all the possible conformational changes between these among these states. And we have folding, we have formation of the unfolded, the protein synthesized on the ribosome, so it exits as an unfolded state, and then we have folding. Other conformational changes can occur as a consequence of functional oligomers, ligand binding, covalent modification, and so on. And we have also formation of native-like states, and so on. And we have turnover. But this is only part of the story, because we also have formation of aggregates. The initial aggregates, you can see they retain some of the structural memory of the initial monomeric state that have originated them. So if we start from a partially folded state, we have a partially structured aggregates, or totally disordered aggregates if we start from a folded state, or native-like aggregates. But eventually, these can develop in different ways. We can have formation of beta structure aggregates and amyloid fibers, but also native-like deposits or amorphous deposits. All these different deposits have been observed in different diseases. And so we started our investigation by trying to address this question. What structural elements present in the aggregates are responsible for cell dysfunction? And what is the mechanism of cell dysfunction? Of course, it's a very ambitious question to answer. And we took advantage of 3 protein systems in our lab: HypFN, alpha-synuclein, and the amyloid beta peptide. Alpha-synuclein is associated with Parkinson's disease. The amyloid beta peptide is associated with Alzheimer's disease. HyperFan is a model protein, but eventually it turns out that this forms aggregates that can reproduce all the effects that have been observed with the amyloid beta peptide. And in particular, we took advantage of the ability of these three protein systems to form different types of oligomers, but where only one type is toxic. So we could generate toxic oligomers of HyperFan and non-toxic oligomers of HyperFan. And later on, we could also generate toxic and non-toxic oligomers of alpha-synuclein. And this is not our work. It was done in Peter Tessier's lab, but we can also generate toxic and non-toxic oligomers from the amyloid beta peptide. And this is very good, very advantageous, because we can clearly start a comparative study between these two species and trying to understand why only type A oligomers are toxic, why only one type is toxic and the other one is not. So what special have these toxic oligomers? Why do they cause cell dysfunction, and why the others do not? And we can just start a comparative structural and functional study. And I'll show, for the sake of time, I'll show mainly results from HyperFan, because this is what we started from, and we have most of our data are on HyperFan. But we could get very similar data on alpha-synuclein and the amyloid beta peptide as well. So for HyperFan, we could form these toxic and non-toxic type oligomers, that we called type A and type B initially, and they are very similar in terms of morphology. You can see that they share a similar morphology, which is spherical or discoidal, and the height, the size of these oligomers is very similar. In terms of secondary structure, they are dominated by a high content of beta sheet structure, as you can see from this peak, this negative peak in the second derivative FTIR spectrum. And they both bind thioflavin T, but very weakly relative to amyloid fibrils. And you can see this is free thioflavin T, and the binding is very small compared to amyloid fibrils. And so the two oligomer types share a similar size, content of beta sheet structure, and they have a similar weak thioflavin T binding. But only type A oligomers are toxic. And to cut a long story short, I can just go through a summary where we showed that type A oligomers are cytotoxic to neuronal cell lines. We started from neuronal cell lines like SH, SY, 5Y, and we found a decrease of MTT reduction in mitochondria, an increase of intracellular calcium, an increase of intracellular ROS, release of calcein due to membrane leakage, and we also have an activation of the caspase-3 as an indicator of apoptosis. Then we started to work with primary neurons, and we found that the oligomers co-localize with synapses, very similar to A-beta oligomers. They inhibit long-term potentiation. They cause a cognitive impairment in whole rats by doing these water maze tests or similar tests. And we also find that they cause a loss of cholinergic neurons in rats' brains, where the loss of neurons is prevalent for cholinergic neurons relative to other neuron types. And again, to cut a long story short, We did structural work using FRET, using solid state NMR and other techniques. We also used Pyren, and we found we couldn't resolve the structure of these oligomers because they are heterogeneous, and it's not trivial to resolve the structure of a misfolded oligomer, but we could find important structural differences between them. First, they are similar in many respects. So they have a similar height in AFM, so a similar size, and they have a similar content of beta sheet structure. However, the toxic oligomers are more structured. They have a structured N-terminus, and you can see the non-toxic oligomers are less structured and they have a flexible N-terminus. And most importantly, due to this rigid architecture, the toxic oligomers expose on their surface hydrophobic residues. And this is very clear using all the techniques that we have used so far. And also we have buried hydrophobic residues in the non-toxic type B oligomers. So we have a different exposure of hydrophobic residues, which appear to be more exposed in toxic oligomers rather than non-toxic. And what is the mechanism of toxicity for type A hyperfan oligomers? Because so far we could use a number of readouts of toxicity, and we could find the cell dysfunction is caused only by type A oligomers, regardless of the readout that we used, regardless of the probe that we used to detect toxicity. And I'll show you a flavor of that. For example, by monitoring the entry of calcium in the cells, we could find only an effect on type A oligomers. So what did we do in this experiment? We used cell cultures. These are basically neuronal cells, and we added the oligomers to the extracellular medium of the cells. So these are the untreated cells with a very small concentration of intracellular calcium. If we add to the extracellular medium the native protein, nothing happens. If we add type A oligomers, again to the extracellular medium of the cells, we have an interaction with the cells, and as a consequence, we have an entry of calcium from the outside, from the extracellular medium, to the cytosol, and this can be detected by a huge increase in fluorescence. Using type B oligomers, we have some effect, but this effect is very small. And more importantly, we could repeat this experiment at different times, and we could determine sort of kinetics with for this entry of calcium, and estimate using a fitting procedure with an exponential function, we could determine the rate constant, which is of the order of milliseconds. Sorry, of 10 to the third second. So not milliseconds, but of course the order of a few minutes, I would say. Then we also probed ROS, the levels of intracellular ROS, and again we did experiments in a similar fashion, so again adding the oligomers to the extracellular medium, and in the untreated cells we couldn't detect many ROS. With the native protein, again nothing happens, and only by adding type A oligomers we could see an increase of oxidative stress with detection of ROS levels. And again, we could repeat the experiment at different time points and get the time constant. And you can see that in this case, the kinetics are slower relative to calcium, in intracellular calcium increase. So showing that the increase of intracellular ROS is slower relative to the increase of calcium. And so we also used additional readouts using a number of biomedical modification and a number of readouts, and we found a number of modifications analyzed and their kinetics quantified. All of them occurred only with type A oligomers, and we could determine the kinetics for all of them. And so we have a cartoon to show what's going on for the toxic type A oligomers. You can see that here we can see the most relevant structural characteristics of them. We added them to the extracellular medium. So this is the cell membrane. This is the extracellular space and the intracellular space. And by adding them, we have an interaction with the membrane. And this is the first event that can occur, interaction with the cell membrane. And we also have a penetration of the cell membrane. And as a consequence of this penetration, we have a destabilization of the membrane. And these are the calcium ions, which can penetrate through the holes formed by the oligomers. And we could estimate the rate constant, and this occurs very quickly. This is actually the higher rate constant that we could estimate in all our readouts. And following the entry of intracellular calcium, we have an increase of ROS, and this occurs slower on this time scale. And again, we could determine the rate constant. And then we have also an internalization of the oligomers, because at some point we can find these oligomers inside the cells, and this occurs more or less on the same time scale as the increase of ROS. Eventually we have measure holes in the membrane, and we have an exit of calcine, and so leakage of larger molecules from the cytosol to the extracellular space, and this can occur with this rate constant. And eventually we have caspase-3 activation, and the cell is more or less rooted to apoptosis. And this occurs on an even slower time scale. By looking at the non-toxic type B oligomers, again, I summarize the main structural characteristics here. And we have an association of the membrane. Actually, we could find that they can also associate with the membrane, but we have no insertion into the membrane and no membrane destabilization. So these non-toxic type B oligomers are still able to interact with the membrane, but there is no possibility for them to associate intimately with the membrane, so to dig into the membrane and to cross the membrane. The interaction is very superficial, occurs on the external side of the membrane, and so the membrane is not destabilized, and we cannot have an entry of calcium, we cannot have a destabilization of the membrane, and any of the biochemical modification that follow the entry of calcium. And this is a picture, a similar picture for alpha-synuclein oligomers that we published 3 years ago. And in this case, in a collaboration with Alfonso De Simone, who was at the UCL at that time, we could determine structural differences between toxic and non-toxic oligomers. Here we can see the non-toxic oligomers, which are which do not have any solvent-exposed hydrophobic residues. They can interact with the membrane, but the interaction is only on the surface of the membrane. For toxic oligomers, actually we have a higher structure in some portion of the protein. Particularly, we have a well-developed beta sheet structure. We also have a flexible N-terminus, and the N-terminus can bind and anchor the oligomers to the membrane. And following this anchorage, we have an insertion of the oligomers inside the membrane. Actually, the structured beta sheet core can get into the membrane. And again, we have the entry of calcium and all the biochemical modification that we have seen. Okay, so we can try to conclude some part of the story and then going back to aging and see how we can interpret this data for aging. Actually, the first conclusion that I would like to draw from this talk is that toxi-oligomers appear to have a more rigid architecture that leads to exposure of hydrophobic patches able to interact with biological membranes. This is something particular that occurs for these oligomers, because when the oligomers are flexible, actually they can bury the hydrophobic residues inside. So the more toxic oligomers are those that are non so rigid. The more dangerous oligomers are those that are non so flexible, because if they have some rigidity, they cannot bury completely their hydrophobic groups inside. They leave some of the hydrophobic groups exposed on the surface, and these allow them to interact with the membrane to cause all the cell dysfunctional phenomena that we have seen. A calcium influx across the membrane appears to be the earliest event in the toxicity cascade, and all the other follow this event. This causes other cellular dysfunctions such as ROS accumulation, mitochondrial dysfunction, cell leakage, etc. And so we started from a picture like this, and so all the dysfunction, all the loss, the loss of function that we have in aging, like the loss of proteostasis, but not just, causes the accumulation of misfolded proteins, fibrils, larger aggregates, deposits, inclusion, oligomers, whatever you like to call them. And as David Smith said, the oligomers are probably the most important and the most deleterious part, the most deleterious species in protein deposition diseases, and they form as a consequence of a loss of power from those protective mechanisms. But this is only part of the story, because the accumulation of aggregates can also increase all these phenomena. We have just heard from David Smith that actually these oligomers can make the UPS system less functional. And so there is a vicious cycle here where the oligomers accumulate as a consequence of loss of proteostasis, but the accumulation of oligomers itself can inhibit further the loss of proteostasis, and we have seen that they can accumulate ROS, we can have mitochondrial dysfunction, and all these, we can have less cell loss because eventually the cells die. So the oligomers can cause a number of effects that maybe determine their appearance in a sort of vicious cycle where the cell cannot escape anymore. And let me just end by thanking all the people involved in this work over the years, and thanks also to external people involved that are not in my group, not in the University of Florence, and involved in this work. And eventually, let me thank my group for all this work. Thank you very much.